Hello, everyone, and welcome to the latest MDPI webinar on cells, which is entitled Molecular Mechanisms of Exercise and Healthspan. Thank you very much for joining us today. This is the sixth webinar of a series that is based in special issues and sections from our Cells journal. So if you're interested in submitting your manuscript to the special issue related to this webinar, please check the webinar's webpage. Our chair today is Dr. Robert Wessells from Wayne State University School of Medicine in Detroit, the States, and we will have five speakers presentations. We will have also 2 time slots for the discussion, and so we will address all the questions submitted by live online audience. So please, if you're joining us live, type your Q&A box that must be below on the Zoom screen. And please mention the name of the speaker together with your question, as we have more than one today. I would like to remind everyone that this webinar is being recorded and that the recording will be posted soon on the webinar's webpage. Now I'd like to introduce our chair. Dr. Robert Wessells is an associate professor of Wayne State University School of Medicine in the States. He acquired a PhD in molecular genetics from Ohio State University in 2000. He received postdoctoral training in fruit fly genetics and physiology at the University of Michigan and the Burnham Institute for Biomedical Research before taking his present position in 2006. His research interests focus on cardiac senescence, effects of exercise training on functional aging, insulin TOR signaling, and fatty acid transporters. Now I hand over the floor to our chair, Dr. Robert Wessells, and please, all of you, enjoy the webinar.
Okay, thank you so much. I'm really excited to have all of you here, and especially thank those of you from more Western time zones where I know it's really early right now. And the topic that brings us all together is exercise and health span. And for the purposes of this webinar, we're defining that broadly to include how exercise helps elderly people retain greater degrees of function, and also how well exercise can reduce the incidence or the severity of age-related diseases. So what we're all very interested in, it's now well recognized that exercise can do these things, but we're interested in figuring out what are the molecular mechanisms by which exercise can exert these effects. And there are 2 main reasons why we might be interested in that. The first of which might be that it will allow us to generate better, more individualized exercise programs in the clinic for individuals. And the second, and perhaps even more exciting possibility, is that the discovery of exercise-induced molecules that can mimic some of these effects has extraordinary potential to be leveraged for therapy that can affect a wide variety of disease states and also loss of function due to normal aging. And we're going to hear a lot about that over the next 2 mornings of this webinar. And I'm excited about the variety of model systems that are going to be represented over the course of the 2 days, ranging from traditional rodent models, mice and rats, and also newer invertebrate genetic models, not new models, but newly brought to bear on this particular problem, like the fly and the worm, which these short-lived animals also have a lot to teach us about aging and how exercise can affect that. And as exercise adaptations appear to be universal across the animal kingdom, we hope that all of these models across this spectrum will inform us on how things work in humans and what we can do to leverage learning from all of these animals. We're also going to look at a bunch of different areas of study. We'll hear talks about muscle. We'll hear talks about how exercise affects the brain. We'll hear talks about organelles. And in not only the function of organelles in exercising tissues, but also we're going to hear one talk about a mitochondrially encoded gene, which plays a big role. And we'll hear about metabolism during normal aging or during exercise itself. And we'll also hear about how we can leverage some of these things to combat acute and chronic disease states. So a lot of information coming at you. So our first speaker is Nicole Riddle from University of Alabama at Birmingham. And Dr. Riddle has been studying epigenetics for quite some time. And I'm really excited about her recent work using a new Drosophila exercise modality and using it to study variation in the exercise response among different wild types. This work has been very inspiring to my lab, and I'm really excited to hear more about it. And so with that, I will turn it over to Nicole.
All right, well, thanks very much for that kind introduction and for giving me the opportunity to tell you all a little bit more about the work that my lab has been doing over the last 5 or 6 years, trying to understand exercise response variation and using Drosophila to do that. Okay, so just like Dr. Wessells introduced for us, exercise is really an important part of a healthy lifestyle. And many of us use exercise for a variety of reasons, including weight control, to strengthen bone and muscle, to just feel better and have basically more physical fitness. But the medical field also recommends exercise because it has really nice impacts on disease risk and also on several diseases that it can alleviate. And so you're probably familiar with the fact that cardiovascular disease and type 2 diabetes and metabolic syndrome are all things that exercise can help with. You might not know that it also can really help lower your cancer risk and even premature death risk. The medical community particularly likes exercise because there's low inherent risks at the level that they recommend exercise for most of us, which is very different from professional athletes. There's really very little risks of doing exercise. And so that really shows you that it's very, very important and a very good treatment option for a lot of things. And 50% of Americans, for example, meet like the CDC guidelines of aerobic exercise that we're supposed to be getting every week. But there's also obviously another 50% that don't. And part of the reason that we have individuals that probably don't exercise has to do with variation in exercise response. As you probably also aware from your family and friends, not everybody responds the same way to exercise. No matter what exercise response measure we use, be that like changes in weight, changes in muscle strength or endurance, when we look at a population of people on the same exercise regime, we get a variety of outcomes. Some people do really well, but there's annoyingly up to like 20% of people that really just don't see the response that they were hoping for. And that obviously can be really frustrating, and we don't really understand why that is. And so that's the question that I really am interested in trying to understand where is that variability in response coming from? And is there really such a thing as exercise resistance, so people that just won't respond to exercise? And so we are trying to use the Drosophila model to look at the genetic and epigenetic impacts on this exercise response variability, to understand how much of this is genetic or potentially epigenetic. The reason why we think that there is a genetic component comes from a variety of studies. One of them, for example, is some studies in mouse that show that you can actually select for animals that like to basically exercise, that are high wheel running. So they run on the wheel all the time. They spend lots of time on a little running wheel. And you can also select for the opposite direction and get couch potato mice strains. And that's genetically inherited. You can keep these strains going for a very long time. Studies in lots of animal models also have shown that we can have mutations that cause changes to running behavior or general animal activity. So we can have mutations in a variety of genes that cause hypoactivity, so less activity than normal. And we can have mutations that lead to hyperactivity, animals that are, I guess, fidgety and they just move around a whole lot more. And finally, in humans, there is data that suggests that certain genetic variants are overrepresented among elite athletes. And all this tells us that genetics really does seem to be a play in this. And so maybe we can understand what the genetic role is in this exercise response. And we use fruit flies to get at this question. So the first question I always get is, well, how do you exercise flies? And we don't have little treadmills for them. Instead, what we do is we utilize an innate behavior that they all have, which is called negative geotaxis. This behavior means that if you have the animals in a small enclosure, they tend to hang out at the top of the enclosure. For reasons that I don't fully understand, they like hanging out at the top. And so if you manipulate things like tapping the little vial on a hard surface, the animals fall down to the bottom of the enclosure. And then because they want to be at the top, they start running around and running to the top. And so basically we can utilize this behavior in a variety of ways to trick them into working out, basically. We utilize a system called the treadwheel, which was developed by Dr. Laura Reed, who you will hear about from later. And her lab and our lab have basically characterized this system a couple years ago. In this system, we have a rotating rod. Not sure you can see my mouse, but there's the rotating rod. There's some clamps, and with these clamps, we can snap our like flywheels on there, and then we're going to rotate them end to end so that the direction of up changes. And so if you do this slowly enough, the animals will just keep running because they're chasing where up is in the low while. We can show that these exercising flies with this machine leads to physiological changes. We can also see that this leads to gene expression changes, for example, in mitochondrial genes. And we can also show that the outcomes that we see either in the physiological changes or gene expression changes depend on the sex and genotype of these animals, giving us a pretty good clue that this genetics really does play a role. So my lab then basically had another challenge to face, and that is like, how do we actually compare exercise levels? So we put all these animals on this rotating machine, but we're not really sure how much they're working, right? And so my lab built a derivative machine, which is called the REX, where we use an activity monitor to monitor the activity while the animals are rotating. So we have an activity monitor here that's on a rotating arm, and that we can rotate the whole system. In this activity monitor, we have infrared beams bisecting the vial in the middle, and it will count every time an animal crosses the midline. And so we can get a pretty good idea of how much these animals are working. Our basic experimental setup is like this. So we grow our animals under controlled conditions. We separate them out by sex and genotype, let them recover for a while, and then we put them on our REX machine, first without rotation. So we can just measure what is their natural activity level. So we can figure, which we call basal. So this is baseline level of activity, no rotation. And after that, we turn on the rotation and then measure for 2 hours how much they will actually exercise. And we use the DGRP strain collection, which is a collection of inbred wild-derived strains that was established by Dr. Trudy McKay's lab. And they're all been sequenced and genotyped and phenotyped for lots of things. And so they're really good for running genome-wide association studies. Here's the data from this very first experiment, where we have over here the animals with no rotation. On the top, we have the females. On the bottom, we have the males. And you can see that there's lots of variability in these lines about how much these animals move. You can also see that there's a big difference between the males and the females. The males move around a whole lot more than the girls do. When we turn on the machine then and induce the rotation, you can see that the level of activity goes up. So that means we're actually managing to make them exercise. We're making them work more. And this is really obvious for the females, but you also see an increase for the males. And so the activity level really depends on the sex, clearly, on the genotype. You see how different all these lines are, and on the treatment, if we're having rotation or no rotation. Now, if we look at this data in a slightly different way, if we can look at the correlation between what happens when we have rotation versus we have no rotation in the two different sexes separately. So over here, we show that for the females, and we're correlating the activity levels without rotation to with rotation. And we see a pretty nice correlation here. Same for the males. It's a little lower, but we see a pretty nice correlation. So the basal and induced activity levels correlate well with each other. We can also look at how the two sexes compare to each other. And here at the basal level, there's a significant correlation, but it's a low correlation. So at the baseline, because the females seem to move just so much less, we see a lower correlation. This correlation gets much stronger when we turn the machine on, which suggests to us that what the animals are doing, the females are doing at the basal level really doesn't reflect necessarily their ability, what they could do. They seem to choose not to move around much. And once you actually induce them to move, then we see a much stronger correlation between the male and female measures. So now we can take those measures and run a genome-wide association study. With this study, we've detected over 100 genes that are linked to activity levels in control and exercised animals. And I don't really want to bore you with a gigantic list of genes. So I just want to say that we see basically a lot of genes that are linked to central nervous system function. So we see things like axonal growth cone, neuromuscular junctions, things that are linked to the central nervous system and its function. Another group of genes that popped out of the study that was really of interest to us because my lab, the other half of the lab studies chromatin and epigenetics much more in detail, was that we found 2 chromatin genes that were implicated in this exercise activity level. And those were 2 polychrome group genes. And so we validated those 2 genes by knocking them down. And that's here in the middle is the knockdown condition versus on the outside, we have the 2 drivers that get us to an RNA knockdown of this protein. And what you can see pretty nicely is that both for Jared2 and for Susie2, we see an increase in activity levels when we knock those 2 genes down. And so basically this helps validate our GWAS study. So for this first part of the talk, I've shown you that the DGRP fly collection has a very big variability, I guess, in activity levels. We've identified over 100 genes that are linked to activity level in these animals. And interestingly, the genetic variants that are controlling activity level differ between the sexes. We can identify lots of different functional categories, but many of them link to the central nervous system. And excitedly for us, we have 2 polychrome group genes that are impacted here. Now, the next study we wanted to get to was actually looking at these responses. What happens to the animals after exercise? And so we set up a second study, again, with our animals divided up by sex and genotype. This time we have 2 sets. We have control animals that don't get exercised, exercised animals that will receive a 2-hour exercise treatment for 5 days straight. And after that, we assay their physical fitness with a climbing assay, seeing how fast they can climb up on a vial. We assay their weight, so we're weighing them, and then we use that data again for a GWAS to figure out what's the genetic architecture controlling this. Before I'm going to show you that data, I want to just point out a few things. So what I've shown you in the previous couple slides was that we have a lot of variability in the amount of activity we see in the animals without rotation and with rotation. What I'm showing you here is the differential between these 2. What's the change that we actually see in the animals? So a lot of the females, as you could probably guess, the activity levels goes up when we turn the rotation on. But there's a really big range, and we do have some that don't actually seem to move more. In the males, again, we have the majority of them move more with the rotation, but there is, again, some that don't. And so this makes it more complicated in trying to interpret what actually happens to an outcome measure like weight or climbing activity, because we have not the same, they're not all running around the same way. They're also not running around the same way in terms of the pattern of running. What I'm showing you here is the fraction of activity in the first 30 minutes. So the first 30 minutes, if they were running steadily, we would expect them to run, have 25% of the activity there. But that's not what we see. 25 would be about here. There are some that do that seem to be pretty steady in the way they're running, but the majority of them run a lot in the first 30 minutes, and then they just kind of like slow down and seem to kind of hang out. So there's big differences in how they actually respond to this stimulation of exercise, which we need to keep in mind when we look at what happens to things like their weight and their climbing ability. So here is what happens, what differences we see in weight between the control and exercised animals, again, separated out for males and females. And so we have animals that gain weight, and we have animals that lose weight, and we have animals where nothing happens for both the males and the females. And so you can see pretty clearly that what happens here in terms of weight change with exercise, it depends on the sex and the genotype of the animal, as well as the interaction between sex and genotype. If we now relate this back to their activity, so the change that we see, either the change or the increase in activity level that we see with the rotation, this is what we find. So for the females, we see a nice increase, nice positive correlation between how much more they're exercising and what happens to their weight. So these females, we see an increase of weight with exercise. Given what we know about males and females and their body composition, this makes us think that these animals might be converting fat mass to muscle mass, which is basically denser. And so that's why they're gaining weight. Interestingly, for the males, we don't really see any correlation between the amount of activity change, so what their exercise intensity really is, and their weight change. So there's really no relationship here that we can discern. If we now use this data for a genome-wide association study and look at the variety of genes that come out of there, we find that there's a lot of genes that are linked to signaling pathways here. So what seems to be important for how an animal responds to exercise in terms of weight change is very much depending on signaling. So we have signaling Go terms that come out of this. And so that's been really interesting, because that seems to suggest that we need to have signaling coming from between different cell types and from different parts of the body to each other for us to see a change in weight. Okay, so now let's look at a second response measure, which was climbing ability. So we again measured climbing ability in the animals that were and were not exercised, and then looked at the change between the differential between the two groups, which is plotted here as a change in climbing index for either the females and the males. Just like we saw with weight, there's a big range of things that happen. We have animals that seem to get better at climbing. We have animals that seem to get worse at climbing. And we have animals where nothing happens. So again, mimicking what we see in studies of mammals or humans, where we see that there is a range of responses in some animals. Some people just don't respond. If we now do the same analysis and try to figure out how does this change in climbing ability how does this correlate with the exercise intensity? So how much more are they exercising with the rotation? We unfortunately see like absolutely nothing. There's zero relationship between how the activity changed, so how much more they actually walked around, how much more they hopped around, how much more they exercised with rotation, to what we see in this climbing ability. Neither in the males nor in the females, which is surprising, and we're still trying to figure out what this actually means. We've also run, again, a GWAS with this data set, and we find there's really no specifically gene groups enriched in here. So we find some interesting genes. We found another polycomb gene that we're interested in. We have other genes that have been clearly linked to signaling or sensory responses. But there's really nothing in terms of a specific gene group or pathway that we can get to. And so with that, let me summarize the second part of the talk for you. So we basically find that the DGRP fly lines vary immensely in their response to exercise stimulation. So both in terms of how much they're actually exercising, in terms of the pattern of exercise, so when they exercise in our 2-hour exercise period, and then also in if we're seeing changes in weight or climbing ability after the exercise treatment. The GWAS analyses that we've done suggest that there is that signaling is very important for exercise-induced weight change, but we don't see a really clear link between changes in climbing ability and any of these pathways or the exercise intensity that we've measured. And what this means to us is that the exercise responses are really extremely varied and depend on different classes of genes. And that really suggests that it will be difficult to optimize exercise treatments for more than one outcome at the same time, and also that it will be difficult to optimize these exercise treatments for different genotypes, right? Because it seems to be that different genotypes really have such a range of responses that this will be hard to come up with one optimized treatment. And so with that, let me put up the acknowledgements. Most of the work that I've showed you today was done by a graduate student in the lab, Louis Watanabe, who is now a researcher at Riken in Japan, with the help of tons and tons of undergrads, as you might expect, growing all these slides and exercising them. And then here are my collaborators and our funding sources for this work. And with that, I can take questions. Thank you.
Thank you so much, Nicole. I'm going to take the liberty of asking the first question, if that's okay.
Yeah.
I was really fascinated by your data about males and the lack of correlation with weight loss. So in my lab, we use a somewhat different exercise modality, but we saw pretty much the same thing in that the males were not consistently losing weight. But what I'm curious about is, so what we have seen is that in mutants that have extraordinarily high fat, then they do lose weight.
Mm-hmm.
So I'm curious if now that you've kind of weaponized the DGRP set of wild type lines, if you index those according to fat mass, is there enough difference that you could look for a correlation between the stock of fat and whether they lose weight?
That's a really good point, and I'm not sure. So I have not seen a data set that has the fat mass for these guys. If there is, then I should definitely dig that out and do that. So we have done a little bit of analysis using QMR to study fat mass and lean mass. The problem that we're having with these studies is that while with mouse, you can do that without sacrificing the animals. In our case right now, the animals don't really survive very well, so I can't do a before and after, which is really irritating, and we're trying to work with our core facility to get that fixed. But I think you're right. I mean, part of the issue here is really that the males seem to just have a higher baseline level of activity anyway. And so if you're trying to make them move more, there is just really, first of all, not that much of a response. They just, you know, there isn't that much more increase that they're willing to do. And then the second part is that they do have, you know, just in general, a lot less body fat. And so it is, I think, easier to see a response in the females.
Okay, so we have another question from Monica Driscoll. Can you comment on the variation in responses that occur among members of a single line? Is there a hint of epigenetic regulation within individual lines?
Okay, so yes, there's clearly variation within a single line, but we don't have enough data, I think, to really tease that apart, right? Because our measures are on, they're usually either 5 groups of 20 or 10 groups of 10 individuals. And so I don't think we can really say anything about that. I do think that epigenetic variation, I mean, there's definitely some there, right? And so it's difficult for us with the data that we have to address that question, but I think it's an excellent question.
Okay, one last one before we move on to the second talk. From Mike Hudeman, did you see any changes in genes related to mitochondrial energy metabolism, specifically oxidative phosphorylation or mitochondrial biogenesis in the different groups?
We have a couple of genes that are linked to mitochondria, but there was really not very many. I mean, it was literally like a couple that showed up, but it's you know, not like it would be a big group that comes out of it, maybe 1 or 2. Yeah.
Okay, thank you very much. Now it's time for our second talk. So Moh Malek is currently at the College of Pharmacy and Health Sciences at Wayne State University, where he is the director of the Integrative Physiology of Exercise Laboratory. He did his training at University of Nebraska and also University of California at San Diego. And he's going to talk to us today about a really cool and innovative way of trying to protect against ischemia reperfusion injury. Take it away, Moh.
Okay, well, I certainly appreciate the invitation, and I'm actually very humbled because a lot of the panelists are individuals that I look up to in terms of their work and follow. So, you know, being in the same, you know, seminar or talk with, you know, the panelists that I look up to is certainly humbling. What I wanted to do is, first of all, acknowledge that, you know, this work that I'll talk about is really a collaboration between myself and Dr. Hudeman, who, you know, is the mitochondrial sort of expert, if you will. And so it's interesting because, you know, you combine an exercise physiologist with a mitochondrial biologist, and, you know, you get some interesting things that it can occur, and we have a tremendous track record of publications over the last 10 years. So just to give you an idea of today's agenda, so we're going to be talking maybe not so much about exercise, but we're going to be right on the envelope of where this webinar is going to be. And specifically, we're going to talk about total knee replacement, the role of mitochondria in ischemia reperfusion, and then, as Robert said, talk about a novel and innovative approach to reduce the ischemia reperfusion injury. And then at the very end, some points to consider, because the data that I'm going to share with you is unpublished, in preparation. So there are still things that we need to consider. Now, overall, in terms of total knee replacement, you know, one of the reasons that it's done, or a number of reasons, you know, has to do with these here. And it's in individuals who are older than 55. Typically, it is a worldwide, you know, phenomenon. It's not just specific to the United States, although recently there was an article that came out that said it's projected to increase, you know, by 2030, and the cost varies. And this Blue Cross Blue Shield study or article showed that the cost does vary based on where you are in the United States. So I thought that was interesting in of itself. Now, the problem, or what's bringing on this ischemia reperfusion injury, has to do with what is being done in the surgical unit. And so in order to have a clear field, surgical field, the leg is cuffed, it's inflated, and so, you know, the surgeons can do their thing without, you know, having to do blood transfusions and such. Now, there are a couple studies that have looked at the effect of using the tourniquet. So this first one, and I'll just give you sort of the 35,000 foot view of each of these studies. This one essentially concluded that there is no nerve damage, because there has been some arguments in the literature that the cuffing does cause nerve damage. And, you know, this study, you know, found in a large number of subjects that that wasn't the case. This next study essentially found that, you know, the tourniquet does significantly decrease the intraoperative blood loss, calculated blood loss, as well as the operation time. But interesting enough, there was sort of no significant decrease in DVT. And so again, you know, there were some complications, but so the main purpose of reducing blood in that surgical field, they found that met its goal, if you will. And then this last one, which is more recent, and there has been talk in the field to perhaps come up with alternate ways of doing this. The chief surgeon that we work with, they still do this, but they themselves also acknowledge that there needs to be other potential avenues, instead of just doing this tourniquet or the cuff. This particular meta-analysis found that there's an increased risk of certain severe adverse events, pain. But they also found that the tourniquet does reduce the time in the surgical unit, if you will. So, you can sort of get the idea that it's an unintended consequence of having this surgery. You need to have the surgery because maybe now you're adopting a sedentary lifestyle. You can't do activities of daily living. You're dealing with pain, but the unintended consequence is that you have a ischemia reperfusion injury. And so Hans Dreyer has looked at this, and essentially, what they have shown is that you have muscle atrophy that occurs, immediately, if you will, 2 weeks, and then 6 weeks after. So there is some, issues related to having the surgery above and beyond a healthy knee. Now, he has hypothesized that it is the sort of ischemia reperfusion injury, that unintended consequence that then sets off a series of signals that result in atrophy of the muscle. So in terms of the role of the mitochondria related to ischemia reperfusion, I won't go through all of this per se, but the basic take-home message is that you have an increase in reactive oxygen species when you have this ischemia reperfusion injury. And that is linked to, and I'll go to this next slide here, to the membrane potential. And so here you have membrane potential. There's an ideal range for a cell's membrane potential to be healthy, to work as it should. And on either side of this optimal range, if you have ischemia, then you have energy depletion. If you have reperfusion, then you have this excess increase in ROS. And you can sort of, think of this as, a couple of years ago, the Samsung phones that would get charged and then blow up. So that's kind of what's happening here, sort of, in a sort of analogous way, if you will. Now, my colleague, James McCauley, he was kind enough to have me use this slide. And essentially, there's really nothing that's gone to the clinical trials that's worked in terms of ischemia reperfusion injury. Now, this is not specific to the hind limb or muscle. He does a lot of cardiac type injuries of ischemia reperfusion. But the facts are that there really isn't anything out there that works. So this handsome young man right here is Mike Hutterman, and he's too humble to say it, but I will. He is an expert in mitochondrial biology, and he's dedicated his career to COX, which is complex 4. And without going into too much of the specifics, one of the key things that you want to sort of focus on is you have this copper A right here, and you have copper B, and they're strategically placed because copper A essentially is where the electron comes in, and then copper B is the oxygen binding site. And of course, we have, energy production ultimately. Now, what Mike did about, I'd say 15 years ago, was to look at a spectrum from 700 nanometers to 1,000 nanometers. And they developed this device, and they ran, the spectrum to see if the infrared light, if there's any unique features. And you can see here that the 810, that's the common one that you see sometimes in physical therapy back in the days, that actually activates COX, or cytochrome C oxidase. And the 750 and the 950 actually decrease COX activity. And this is critical because COX activity, modulating that, controlling that, you can control membrane potential, you can then control ROS production. So that's sort of the flow of where we're going. Now, they used purified COX from bovine tissue. You can see here that when they used the 750 light, there was a decrease. Here they looked at oxygen consumption rate. This is just COX activity. And you can see it pretty much mirrors what you're seeing. And again, if you do the combo, you get a sort of a double effect, or maybe not double is not the right word, but you get a synergistic effect. And here, this was, I believe, in liver tissue. And you can see that if you just switch it on, the potential goes down, but if you switch it off, it goes right back up. So it works in a switch-like manner, if you will. So here are the electrodes. So essentially, this is the setup when we use it for our animal model. It doesn't generate heat, and I'll show you data on that. And then there's a fan that takes any heat that there may be out and into the environment. So this was a study that they did with stroke. And essentially what they found is that you can reduce ROS activity using this marker. And they did more functional stuff. And the take-home message in all of this was that you can preserve the neurons that's related. And even if you delay the therapy, meaning that you delay it by 30 minutes or 60 minutes, and again, this is global ischemia reperfusion in the brain, that you can still preserve the neurons. And you can see here, the temperature doesn't really change. This is 15 minutes, but we have in our lab up to 3 hours, and it's the same as what this looks like. It does not change. The tissue or the animal isn't heating up or anything of that nature. So, when Mike was showing me this data, I said, you know what, there may be some approach to ischemia reperfusion in skeletal muscle. And so, we came up with this sort of hypothesis, if you will, that relates to what potentially is going on in individuals who are getting total knee replacement. So we started off with using bulb C, and the reason why we use bulb C mice is because C57s are a little more robust, if you will, or tolerant to ischemia reperfusion injury. And this is data from others' lab that they have done and published. We went with bulb C. We used a non-invasive approach, a band that you'll see. And essentially what we wanted to do is look at it in 1 hour. So that would mimic roughly the operation time, and then 4 hours. And the 4 hours is that there are studies that show in skeletal muscle that if you have an ischemic condition in the muscle there's a skeletal muscle, 4 to 6 hours, you're sort of on that brink of killing the muscle, if you will. And so I told Mike, I said, if this works, then theoretically we should be able to rescue the muscle maybe at 4 hours. We haven't done 6 hours, to be quite frank, but the treatment is only 2 hours of infrared light treatment. So you can see here that when we band, we cut off the blood supply. It's about 93% of the blood supply is cut off, and that's consistent with other studies from other labs that look at the band. You can see here clearly what's going on in the leg. And the animals are anesthetized when we put the band on, but then we put them back in their cage and they're good to go for the 1 hour, 4 hours, whatever. And then here is the infrared light treatment. And again, they're under anesthesia. So you can see here that COX activity, just like we thought it would during the ischemia, increases. But 4 hours later, there's not much of an increase. So the initial increase comes up front in the first 1 hour. We looked at the swelling, if you will. So it's the same leg before we put the band on and the same leg 4 hours after the band, with or without infrared light. You can see that the 810, you actually had more swelling relative to the infrared light combo that we used. We also looked at essentially, a very quick way of looking at tissue death. And again, this is 4 hours and then, no light or with light. And you can see here that we were able to, preserve the muscle, certainly much more than just reperfusion alone. We did the mitosoc experiment similar to what Sanderson did, and essentially used the same model, except the dosage was different because they used it in a rat, we used it in a mice. But you do a tail vein injection, wait 30 minutes, and then do your experiment. And right after the light treatment, you euthanize the animals and you go from there. And again, you can see sort of consistent with what we hypothesized. And then we did some functional work related to looking at membrane potential. And again, take-home message is we're able to, control with this infrared light the membrane potential. So a couple of points to consider. I know a colleague one time when I gave a version of this said, well, COX may not be the mechanism, and they referred to this particular article. And the reality is this article is, not accurate because the author doesn't really talk about those copper sites that are only specific to complex 4, and none of the other complexes have it, and that's what's absorbing the infrared light. And Dr. Hutterman subsequently had met this person at a conference, and they talked and sort of convinced them that what he was saying was maybe not accurate in terms of that. And this was just basically a paper, like a hypothetical or a editorial paper, not necessarily empirical research. This other bullet point related to RAWs and, maybe it's not the right situation in terms of what we're hypothesizing. What I would say is this diagram sort of says it best. So if you have a pathologically high mitochondrial membrane potential, this leads to a reverse of the electron flux. The excessive RAWs production then occurs in complexes 1 and 3. I've had some also say, maybe mitosocs isn't the right type. maybe use AMP Red. I think you've seen from all the different ones we used. I mean, the data is what the data is. The infrared light reduces ROS no matter how you look at it. I'm not opposed to using Amplex Red, but if it's consistent with what we're finding with other markers, we're going to find the same thing. And then lastly, this related to atrophy. And again, I think if we control the ischemia reperfusion, we can control everything downstream of that. And then just quickly, I know I've got less than a minute or so, the benefit of our infrared light over like drugs and compounds is that we're not really looking or relying on the circulatory system. It's non-invasive, it's quick acting. It is not a scavenger in terms of what some of the other compounds or drugs may be. And, you know, it does have therapeutic effects, and it can be easily transitioned to clinical trial. Lastly, it is a platform technology. So the initial work has come related to stroke, but there are others that, and that's my time from my own timer. And so that is all for today.
Okay, thank you, Moh.
Sure.
So I'll just take the time to ask, you know, one really quick question. Can you extrapolate for us, or actually, you know what, I'm going to give you Monica Driscoll's question, which is appearing now in the Q&A. Can you comment on how deeply the light penetrates into tissue? Is there a gradient of differential benefit?
Yeah, so actually that that third bullet on the second to last, we actually have published using cadavers, and you know, the penetration is pretty deep. It goes right to, you know, it goes past the skull. We've even used a pig skull, if you will, or to look at the penetration. So it does go deep. You know, Dr. Hutterman, Mike, if you want to elaborate and give a little more detail on how deep it goes, I can turn that over to you.
Yeah, I can quickly comment on this. So in the adult pig studies that we've not published yet, we have shown that we achieve therapeutic doses of the infrared light at a depth of 4 centimeters. And just this week and last week, we conducted cadaver studies in the human brain. So we haven't done that with the muscle yet, but in the brain, where we measured at 5 centimeters into the brain. So this is sort of where we are standing right now with the currently available laser system that we have in the lab. I would say we can realistically achieve, again, with the current setup that we have, down to 5 centimeters.
Okay, thanks, Moh and Mike. We're going to move on to the next talk now. Zhen Yan is actually one of the very first people I met in the field of exercise when I first got interested in this, and he's been a great friend and mentor to me, and sometimes collaborator over the years since then. So I'm especially excited to hear his recent work. Zhen is at the University of Virginia School of Medicine, and where he is also the director of a cardiovascular research institute. And take it away, Zhen.
Thank you, RJ, and thank you for organizing this great webinar. So today I'm going to talk to you about a story that developed over the years, and with the big question, the impact of exercise. And since ancient time, we all know that exercise can improve performance. Most importantly, exercise helps us to keep healthy and prevent disease. The million-dollar question is the mechanism underlying this. I think lack of a mechanism and make the implementation more difficult. And also we cannot take advantage of the scientific reason behind it to develop memetics to help to promote health and prevent disease. So my lab take advantage of two different systems in mice. And one is volunteer wheel running, and we recently developed another model of voluntary weightlifting. These two models help us to address the question, the impact of exercise in various different models to prevent disease and promote health. And as you can see from these different pictures, and really every one of them pretty much from our studies showing the impact of exercise on the whole body system, ranging from the brain all the way to microbiota. So a story that developed over the years started with a very simple question, and that is very well observed in the clinical situation. That is, muscle atrophy occur in most cases under the condition of chronic disease to glycolytic fibers. So the question is why oxidative myofibers are resistant to chronic disease in developing muscle atrophy and catabolic wasting, except to the condition of disused microgravity and the denervation. These are all related to the usage of the muscle. So with that, we started to address the question by using mice and then look at the different possible mechanisms. And of all those possible reasons, and the one very prominent one is the antioxidant defense system is very different. In the oxidative fibers, they have more antioxidant enzymes like SOD and the catalase. This very classical antioxidant defense system is very robust in the oxidative fibers. But during the study, we found another feature which is very interesting. The NO-mediated antioxidant induction is more robust in the oxidative fibers, but literally not there in the glycolytic fibers. So we are extremely excited about this opportunity. So we moved on. And one of the talented postdocs, who is a professor at Nagoya University right now, he came to US. And when I was in Singapore, I recruited him to Singapore. He moved with me to US. And he started with a simple question. If we use a drug that clinically used called dexamethasone, and which is used for a lot of diseases like autoimmune diseases, and we use that quite a lot. But one of the side effects of this dexamethasone is induced muscle atrophy. So Mitsu asked the question, what if we provide muscle with an NO donor? The NO can induce antioxidant genes in fast-twitch muscle. And can we prevent muscle atrophy? The answer is quite obvious. When he injected GSNO through the system, IP injection, before the induction or injection of dexamethasone, it can significantly reduce muscle atrophy in all these muscles. But as you can see, the slow-twitch muscle is a resistance to dexamethasone-induced muscle atrophy. They have much less muscle atrophy. But if you inject the GSNO, you can pretty much eliminate all the muscle atrophy in the soleus muscle and then reduce the fast-twitch muscle to a great significantly. So, and then he asked the question, is this due to the induced expression of genes, antioxidant genes? To our surprise, the only one that is induced by GSNO is EcSOD or SOD3. This extracellular superoxide dismutase is the one induced by GSNO injection. So then the question is whether exercise can induce EcSOD expression, and what is EcSOD? So EcSOD is copper zinc superoxide dismutase with exactly the same structure as SOD1. The one exception is that this EcSOD has a signal peptide that will direct this secretion from intracellular space to the extracellular space. It's homotetramer, and it can travel in the circulation. It can also bind to the heparin sulfate proteoglycans to attach the cell surface. It depends on how much heparin sulfate proteoglycans you have, it may have high affinity to certain cells. And in the C-terminal region, there are 6 amino acids. They are all positively charged, R, KK, RRR. This called a heparin binding domain is the one is responsible for EcSOD binding to the heparin sulfate proteoglycans. So this is very critical. This is like specificity for binding. So this EcSOD is unique. It's unique. It's secreted in the extracellular space. It's unique. It has this binding domain. So Mitsu did one study first, asked the question, if we collect the muscles, that different muscles like this white vastus lateralis muscle here on the superficial part of the thigh, or soleus muscle here, can you see the difference? The difference between these two muscles, if I use a human muscle as an analogy, if I stand here, I need to use soleus muscle to support my posture. The vascular lateralis, or the superficial part, or white vastus lateralis muscle, is the one you use for 100-meter dash. How many of you have run 100-meter dash in the last week? Probably very few, or maybe none. So we don't use this muscle very much. So this is the locomotive muscle. This is a phasic muscle that we rarely use. If you see the EcSOD expression is highly expressed in soleus muscle, less expressed in the white vastus lateralis muscle. He confirmed it by immunofluorescence. As you can see, another muscle called plantaris muscle is a mixed muscle, less oxidative ones. You can see the more expression in the soleus muscle than the plantaris muscle. You can also see the enrichment in the aorta and the lung tissues, extremely highly expressed. But if you look into the immunofluorescence, you notice that around each muscle fiber, you see those dots there. If you enlarge the image, you start to see this circular structure. These are the capillaries. So actually, EcSOD is expressed in skeletal muscle, but secreted, it can bind to the surface of the endothelium. So this is one feature due to the presence of that heparin binding motif. So the next question Mitsu addressed is, can exercise induce the expression? He used the volunteer running. We took the plantaris muscle, which is recruited during the running. He detected significant increase in the plantaris muscle after 4 weeks of exercise training. Then he asked the question, is EcSOD sufficient to provide protection against the muscle atrophy? And then he used a model of electroporation. And this is a very effective way to do somatic gene transfer with a single injection of plasmid DNA followed by electroporation. He got a 60 to 70% transfection efficiency with co-injection of GFP, so we can detect the positive fibers. As you can see here, Western blots show very profound induction of exogenous EcSOD. And then he measured the positive fibers in response to dexamethasone injection. As you can see here in the wild type animal without injection of EcSOD, but an empty vector, you can see this loss of muscle fiber size, but it's preserved in the positive transfected cells with EcSOD. So that's answer the question, EcSOD expressed in the muscle can protect the muscle fibers from atrophy. But since EcSOD is expressed secreted into the circulation, we asked the question, can we induce the protection against heart failure induced muscle atrophy? So this is still in the skeletal muscle, but use a different model. This is a genetic model of heart failure. And we made a transgenic animal by using MCK promoter, expressing EcSOD or SOD3 with a flag tag in skeletal muscle. As you can see here in the soleus plantaris white vastus lateralis muscle, more expression of EcSOD, particularly in the fast twitch muscle because the MCK promoter is more active in the fast twitch muscle. And then we put them in the genetic background of CSQ mice, as you can see here, the muscle atrophy is more prominent in the fast twitch muscle than the soleus muscle in the heart failure model. And then in the transgenic animal, less atrophy. Look at the body weight is an indication of cachexia. The CSQ mice had a significant reduction, moderate rescue by the transgenic overexpression of EcSOD. When we put these mice on the treadmill, one thing we notice that is relevant to humans is 60-minute walk. And this one, we did the treadmill running endurance test. As you can see, the running distance dropped from about 3 kilometers to about 700 meters. And in the transgenic animal, just because of the EcSOD expression, they can have significant elevated running distance in the transgenic animal, even though their heart failure was not improved. And then we confirmed there was no improvement of the heart function. So another postdoc, so take a village to do the studies. Another postdoc, Jared Call, who is a tenured professor at the University of Georgia now, and he came and he asked the question, can the EcSOD travel to the heart? And the answer is yes. Look at the heart expression after exercise training. In the serum, exercise trained mice has more EcSOD. There's a quantification of the data, but you look at the EcSOD mRNA, not elevated in the exercise trained heart. So the EcSOD is coming from skeletal muscle after training. He also took advantage of the transgenic animal. Look at this serum level of EcSOD is 6-fold higher in the transgenic animal. In the heart, at least 2 or 3-fold more EcSOD. No induction of EcSOD gene in the heart due to the transgenic expression. So both exercise model and the genetic model show the elevated EcSOD level in the heart. So his next question is, what happened to a model of diabetic cardiomyopathy? He injected STZ to induce type 1 diabetes. This is a very high dose of STZ, kill all the pancreatic beta cells, and it induced hyperglycemia. But then 10 weeks later, you start to see this dilated cardiomyopathy or diabetic cardiomyopathy. And in the transgenic animal, cardiac function is preserved. And you see the fibrosis with STZ injection, less fibrosis in the transgenic animals. Then he took the heart and measured protein and mRNA. You can see the clear evidence of protection. Phospho P38 miles calcineurin induced in the STZ mice, but reduced in the transgenic animals. Look at these ANP, BNP are both classical markers of heart failure, significantly reduced in the transgenic animal. Inflammatory markers, TNF-alpha, IL-1, all reduced. So we had a working model showing that EcSOD can be induced in the skeletal muscle and travel through the circulation, bind to the endothelium and even endocytose into the myofibers, provide protection against oxidative damage to the myocardium and diabetic cardiomyopathy. I'm trained as a surgeon and with a medical background. So my question has always been medical relevant. So another physician came to my lab, saying that he wanted to do an internship before he moved on to his fellowship. So he said, this is a great project for me. And he did that. And by taking the transgenic animal, measure EcSOD, as you can see, this is a muscle-specific promoter, highly expressed in the skeletal muscle. But then in the white adipose, brown adipose tissue, liver, heart, and the lung tissue and the kidney, he saw elevated EcSOD, suggesting that transgenic expression of EcSOD in skeletal muscle can lead to the elevation in different tissues. And we found in beta cells too, pancreatic cells too, but not in the brain. This data suggests that EcSOD may not be able to travel and across the blood-brain barrier. The blood level of activity increased by 6-fold. So with a medical background, we asked the question, what about a severe disease condition called a multi-organ dysfunction? This is the disease relevant to antioxidant defense system in all tissues, pretty much, vital organs. So like in the ICU, 80% of the patients died of multi-organ dysfunction. So we asked the question, if we mimic multi-organ dysfunction by injecting LPS endotoxemia, we kill 100% of the mice. What happened to the transgenic mice? To our surprise, 50% of the mice survived with the same dose of 20 mg per kilo LPS. We use clinical markers to measure the multi-organ dysfunction, the bond assay, LDH, creatinine, and liver damage marker. You can see the damage to all organs, but in the transgenic animal, significantly reduced. So that's very exciting for us. We saved 50% of the ICU mice. The question is, what is the underlying mechanism? So we moved on and look at the VCAM expression and the EcSOD expression. As you can see here, EcSOD in the wild type animal, because of the level is relatively lower, you don't see much of expression in the endothelium of the lung tissue. But here in the vasculature of the transgenic animal, you start to see this EcSOD tethered to the endothelium. Then we performed this experiment. This is parabiosis. You can fuse these two animals with the surgery, and then you can see the wild type animal has a lower EcSOD, transgenic has elevated EcSOD. If you fuse them 10 days later, you measure serum level, you can see the wild type animal receive EcSOD from the transgenic animal. And the control, wild type fusion, did not induce EcSOD expression. Then we injected the pair with the same, double dose. Basically, you inject one animal, another animal, and then with the same dose of LPS. And then we saw that when we took the lung and the wild type animal, separate animal, the transgenic animal, lung are protected, and the wild type animal got this edema, caused this lung sink to the bottom of the tube. But the wild type transgenic fusion lungs are protected. Wild type fusion lungs did not get a protection. Morphologically, we confirmed the collapse of the air space in the lungs, but the preserved airspace in the wild type animal after LPS injection if they are fused. So a technician, Mei Zhang in the lab did this beautiful work. And we moved on to the VCAM expression. In the wild type animal injected with the LPS, you start to see this expression of the endothelial specific adhesion molecule called a VCAM. You can see the expression here, elevated expression compared to the wild type with the saline injection. But if you do that in the transgenic animal, this VCAM expression is significantly reduced. So Dr. Chai from Nanjing Medical University, a doctor in emergency care, and she did the tissue culture experiment with the endothelium monolayer and induced by TNF-alpha to see this VCAM expression. But if she pre-incubated with a transgenic serum, she can block the TNF-alpha induced VCAM expression. So this data is very exciting. It suggests that EcSOD is sufficient to inhibit endothelium adhesion molecule expression. So as I said, it took a village to do this. Another doctor from China, Dalian Medical University, and he asked the question, can we use a blood transfusion experiment like humans? So we took the transgenic serum, injected into the wild type animal, or we injected wild type serum to the wild type animal. So here, wild type animal receiving wild type serum, you see just moderate elevation because there are more EcSOD. But if we inject the transgenic serum, which has 6-fold more EcSOD, as you can see, the wild type start to show the blood level of EcSOD is significantly elevated. And then we used a technology that developed here at the UVA called a microbubble ultrasound. We conjugated a microbubble, which is smaller than the red blood cells, can behave like a red blood cells travel in the circulation, conjugated that with the anti-VCAM antibody, which can recognize the VCAM expression as a way to assess VCAM expression in vivo. So this is the kidney. After 12-day injection of this microbubble, use ultrasound, we can see 10 minutes later, this bubble, a minute later, start to show this bubble. It's beautiful and very bright. And then they disappear, but there is a little bit of retention. That means this is a baseline VCAM expression because the bubble has the anti-VCAM antibody. But then with the LPS injection, you can see the retention is significantly elevated. And then after injection in the transgenic animal, we do not see as much retention, suggesting there is a less inflammation, less adhesion molecule expression. So this is the data probably suggests that why EcSOD is protective, because you can make the endothelium cell less inflammable. And so the immunocells cannot interact with the endothelium cells. Exaggerated interaction may cause the migration of the inflammatory cells into the interstitial space and damage the organs during the severe disease conditions. So a PhD student in biomedical engineering, he took advantage of another system, which is the window chamber, and used the immunolabeled monocells to observe the traveling of the inflammatory cells in the blood vessel. And he found that with the LPS injection, you start to have increased adhesion of these monocells. And this monocytes, fluorescent labeled, they have a behavior. The behavior is that normally they travel along the red blood cells, but they interact with the endothelial cells. They talk to each other. So there is a rolling. The cell stick and then start to roll, travel slowly. But if you have LPS injection, you only have adhesion, but you don't have rolling, suggesting that the monocytes travel and see the endothelial cells with the adhesion molecule expression like VCAM, it was stuck right there. So no rolling. But in the transgenic animal, the rolling is preserved. So putting all these together, we suggest that EcSOD can travel through the circulation to the vital organ vasculature. It can bind to the endothelial cells or maybe internalize through endocytosis. So this EcSOD on the cell surface or intracellular cells can antagonize the superoxide generated during the process of exaggerated inflammation under the condition of sepsis. And this inflammation and interaction of the monocyte inflammatory cells with endothelial cells is the very first step that inflammatory cell can migrate into the interstitial space, cause organ damage. So the last piece of data is relevant to humans. So in humans, we know there are SNPs that cause this mutation of the R to G. This is called a R213G SNP. And Dr. Wessells at the University of Colorado generated this knock-in mice. And so we took these mice, asked the question, what happened? Because in humans, this SNP is associated with poor prognosis of ischemia, cardiac disease, and many other diseases. So we asked the question, what happened to EcSOD? The first thing we noticed in the serum, EcSOD level, normal level here, but in the knock-in mice, you have more EcSOD. Why? Because the attachment to the endothelium to the peripheral tissue is significantly reduced. Look at the kidney, wild type animal, knock-in mice. And look at the liver, not much of a difference. But in the lung, high level of EcSOD in the knock-in mice, significantly reduced. In the heart, in the skeletal muscle. So this is like if you look at the system, the whole level of EcSOD are exactly the same because it's coming from the same gene. Just because of this mutation, now this Patriot missile cannot station somewhere. It's driving around on the street, not providing protection to the strategic target. Now, what happened? If we subject the wild type mice to vesical ligation puncture, which is like a appendicitis with a rupture. This Dr. Chen did this work, and we saw no inflammatory cells in the bronchial alveoli lavage fluid. And so they don't have ARDS. But in the knock-in mice, the appendicitis caused ARDS. And with exercise training, you can only partially reduce it. And multi-organ dysfunction in the same model, showing injury to the kidney and the slight reduction here. And in the knock-in mice, more injury and not much of a benefit of exercise. So this data preliminary suggests that EcSOD binding to the endothelium is critical for protecting the vital organ from damage. Particularly relevant is this ARDS, this bronchial alveoli lavage. This data suggests that EcSOD normally protect the lung from ARDS. Exercise training may increase EcSOD expression and provide further protection. But under the condition of the SNP, you have a vulnerability to ARDS, and exercise can only partially protect. And so maybe exercise, if with the normal, the structure of EcSOD can provide a protection. So we argue that EcSOD and exercise may be one way to protect patients like COVID-19, which cause a lot of patients suffer from ARDS and the death. And so we argue that this is one of the mediators. So with that, I would like to summarize. Endurance exercise promote EcSOD expression in skeletal muscle with a redistribution to peripheral organs enriched at the endothelium. Enhanced EcSOD protection can provide a protection against the muscle atrophy, diabetic cardiomyopathy, multi-organ dysfunction, ARDS. And the mechanism is reducing inflammatory cell endothelial interaction and endothelial activation. Mice with this SNP are vulnerable to ARDS and multi-organ dysfunction and lost protection due to impaired EcSOD binding to the heparin sulfate proteoglycans. And EcSOD is a very good example of the molecular transducer of health benefit of physical activity. And we found more and more evidence EcSOD can provide protection. And then you can search the literature. We wrote a review article. And then after that, I found EcSOD can even provide a protection to the neurodegenerative disease, although it's questionable whether the EcSOD can travel from the skeletal muscle, but does not exclude the possibility that exercise itself through the efferent neuronal input can induce EcSOD expression in the brain. So with that, I'd like to thank the people in the lab. Without their hard work, we would not be able to do this. I also like to thank the funding agencies, particularly Dr. Russell Bollers from National Jewish Health in Colorado, Denver, Colorado, and many of my collaborators here at UVA and my lab here. So with that, I'd like to answer questions. Thank you very much.
Okay, thank you so much. The fantastic story. So I'll go ahead and ask the first question, and I'm interested in the timing. So it's very impressive the way the serum from the transgenic mice has such strong protective effects. For example, when you co-inject LPS. So I'm curious, have you tried looking at a model animal that has already undergone a lengthy pro-inflammatory chronic disease state and tried injecting the serum? Can it only protect against future damage, or can it actually even sort of reverse some damage that's occurred?
Yeah, we haven't done that. I would love to do that. The amount of blood we can collect, so we are collecting the blood. We have the transgenic animal here. We're collecting the blood, and I would love to do that. At this point, I don't have the funding, and I applied. Actually, one of the things, it's exactly what you are talking about, is with collaboration with UNC, with a UNC group, a COVID-19 group, and I propose a proposal to do this injection in the COVID model. And that's one of the prominent one. I would love to do that since the serum is precious and it takes some time to collect the blood, and it's the right experiment to do. Yeah, to do that in a very important disease model and in the treatment manner, because we cannot inject this before the disease. We don't have the luxury to do that. Yeah.
Okay, question from Monica Driscoll. Can you comment on the molecular processes by which the EcSOD is secreted? Does it have to travel through membranes before reaching circulation, and ideas on how it might work inside cells after endocytosis?
Yeah, that is not my particular strong area. I do not know precisely the secretory pathway. It has this, I assume this is a conventional secretion pathway from the Golgi and through the secretion peptide and to the extracellular space. In terms of endocytosis, there are studies. It's not. It's not one of the mechanisms, but the other mechanism, endocytosis, because of the structure is exactly the same as copper zinc SOD. So I assume it's the functionality is the same.
Okay, well, one more question from Mike Hudeman. Have you tried injecting overexpressed and purified EcSOD in the MODS model or in another model?
No, we yes and no. The data we presented with the kidney is the multi-organ dysfunction model, but we have not tried other models. We have not. The purified EcSOD, there are some studies and which almost killed the field. In late 60s, Johns Hopkins performed a study using recombinant EcSOD in human patients during angioplasty. So they have the myocardial infarction. Before they open the artery, they did IV injection of EcSOD and try to provide a protection. At the time, we knew little about EcSOD. We assumed this is just an antioxidant in the blood can have this magical effect. The reality is that the recombinant EcSOD is coming from bacteria, does not have the correct modification, and they injected just minutes before the surgery, or the longest of probably hours before surgery, and then they also have to use a heparin in these patients. So the heparin destroyed the binding, and the incorrect modification prevents this binding, and the injection too short before the surgery did not allow any possibility for the EcSOD bind to the endothelium to provide a protection. So all this combined, I think it's responsible for the failure of the clinical trial. And later, a lot of people just lost confidence in this, say EcSOD has no use. But I never believed that, because I don't believe our body, the EcSOD level we detected in the blood only took 2 microliter of serum to detect that. So, and I never believed our body produced something of no use.
Okay, just a scheduling note. So we're a little bit behind schedule, but I do want to go ahead and have a short break before the second session. So if you don't mind, we'll just adjust our schedule by 15 minutes, and we will start the break time now. So you can continue to ask questions during the break to any of the speakers from the first session, and we'll come back with our next speaker at 15 till the hour. Whichever hour you are at in your time zone, it will be 15 minutes till the hour. So here's the first question of the break time, which we are now in. Is recombinant EcSOD available? I sort of just spoke to that.
Yeah, the recombinant EcSOD is available not at the level of therapeutics, I think. The other thing is that there are companies making SOD, not necessarily EcSOD. there is a company called Preferred Commerce. They do this encapsulated SOD, and a French company is purifying SOD from melons. They use that in drinks and in pills and drinks, and they swear that it's very protective. A lot of athletes doing training, and they have poor recovery from very heavy training. When they use that, they have much better, performance and the recovery. So, but I remain skeptical because I would say promising and skeptical that I think, if a general population with a healthy situation, more EcSOD may be health beneficial, but without targeting to the specific tissue and the organs, but also high level of antioxidant into the gut system, it may have some side effect. You never know, but nevertheless, that's the situation.
So you mentioned at the very beginning of the talk that one of the things that got you interested were the side effects from dexamethasone, which is being used a lot with COVID patients during the last year. Coming back to that, how do you see in the future? Do you imagine that at some point it will become possible to maybe inject some EcSOD to patients who have to go on dexamethasone?
Well, that's certainly the one direction, but that's the translation we need to have from the bench to the bed. And yeah, I think the company with that purified SOD, that with encapsulation with less interaction to the microbiota, I think it's potentially useful because you're going to elevate the level of SOD in the blood, may help, maybe helpful to the peripheral tissues and the organs. So I think that's helpful as long as it's not producing, very bad side effect.
Okay, thanks again to all three of the morning speakers. Those were all very interesting and exciting talks. And right now we're going to be on break until 1:45. And if the speakers happen to see any more questions pop up in the chat, please feel free to either answer in the chat or just unmute yourself and go ahead and answer either way. And at 15 till, I'll go ahead and introduce the next speaker.
Moh, I have a question for you. Dr. Malek, I have a question for you.
Sure.
Hey, how are you doing?
Good, how you doing?
Beautiful presentation. I like your model.
Oh, I appreciate it.
So I do have a question. You present somebody's work in stroke model, and when they use the infrared light after reperfusion, they still saw the benefit, which is a little bit kind of surprising to me, because ischemia reperfusion, really the damaging part is the minute you reperfuse, theoretically, right?
No, that's actually true. I mean, I don't know if it's theoretically, it's actually true, because that's one of the discussion points that reviewers, or when myself or Mike, and Dr. Sanderson is a colleague of ours, on this infrared light, and he's more of the brain person, so to speak. And that is one of the questions is, well, you give the infrared light soon as you start the reperfusion, but that may not really be the case in the real world, especially if you're talking about, let's say, in a sort of emergency situation. And so that's why they also looked at it in terms of a delay. So if you delay it by 30 minutes, 60 minutes, you still are able to preserve some of those CA1 neurons relative to just reperfusion without the infrared light. And then the other unique aspect about that study is that when they use the 810 wavelength, which stimulates COX activity, they saw that things got even worse in terms of the loss. And so that is one of the things that now in this model, sort of with the surgery, when we've talked to the surgeon, we've said, well, probably the best way if we were ever to get to a human model is once they release the cuff, and as they're wheeling them into the recovery room, we would have some type of a infrared light blanket, if you will, that they would just put over the leg and go from there. But have we done those studies where we've delayed it? We haven't. We obviously in the brain it works, but is that the same for the muscle? We don't know yet, but the ultimate goal with this infrared light is to get it to a situation where the footprint is like an AED device that you see like by the swimming pool or somewhere, and then to have it on first responders' vehicles, if you will, so that when they come to the scene, once the patient is stabilized, they can, use the infrared light for, whatever the purpose may be. And certainly we're not saying that it's this is it and you don't need any medication, any other stuff. That's not what we're saying. We're just saying that because, like you mentioned, the reperfusion, it's within seconds that you have this, hyperactivity, if you will, of the mitochondria. What can we do to stunt that quickly? And so the infrared light seems to work, and it also seems to work even if you delay the treatment, at least in that stroke model. So,
yeah. So we did a study that with exactly the same model,
cool.
tourniquet, but we inject Michael Murphy's invention. It's called a mitosnow. It's a mitochondrial NO inducer. And so if we inject the mitosnow right before release of tourniquet, it's very protective. But if we inject it 5 minutes after tourniquet release, no protection.
Interesting.
So, yeah, that is very, to me, I thought, never to be that clean, but that data was quite clean, that you inject too late, it's not working. But it does, these are probably different slightly different mechanism.
Yeah, and that's one of the things is with some of the, as I mentioned sort of quickly in those last couple of slides about, the drug or compound, is that you may have, you're dealing with things that are out of your control. And, you certainly can, some studies do prior sort of priming where they may inject the compound and then do the ischemia. Okay, well, that's not the real world. As you said, with what they do during ischemia, they inject it. It's protective when you have the reperfusion, but if you wait 5 minutes, it doesn't work. So, so again, I mean, we're still working on things, at least when it comes to the leg, but hopefully someday we'll get it funded.
Okay, welcome back from the break, everyone. So we have 2 more speakers in the second session, and the first of which is Karyn Hamilton from Colorado State, where she is a professor and director of the Translational Research on Aging and Chronic Disease Laboratory in the Department of Health and Exercise Science. And she's also an associate director of the Columbine Health System Center for Healthy Aging at Colorado State. So Karyn has been a real leader in applying exercise biology to long-term consequences of aging. And her lab has studied many aspects of the interaction between exercise and aging, including mitochondrial function and turnover, and excellent work on proteostasis. And today she's going to talk to us about some pharmacotherapies.
All right, here I go. So like Dr. Malek said, it's really humbling and a true honor to be amongst the panel of people that are speaking today. So I feel really privileged to be here. RJ already went through my affiliations. This is kind of pictures of where I live. And my plan is to I pulled together a story that is bits and pieces of 3 to 4 different projects that collectively reflect our interest in asking the question, if we have some really good solid evidence that 2 different interventions extend health span independently, what happens if we put them together? Is it always a better intervention, or are there some things that we just need to be concerned about? And then if so, how do we figure out what those are so that we can kind of personalize recommendations that we make to people? So I obviously don't need to take care of this first bullet of my plan here. I think everybody here is convinced that exercise is indeed a health span extending intervention. So I'm just going to split the short time that I have between talking about interactions of exercise with NRF2 activators. I'll move on to interactions with metformin, and then finish up with some newer data looking at interactions with SGLT2 inhibitors. So since I was a trainee, which was just more than a few years ago, I've been really interested in all things redox balance. And it's fun to kind of look back and realize how far we've come from the days where we thought that oxidants were merely things that damaged DNA, proteins, and lipids and other macromolecular components of cells, and that fixing the problem was as simple as throwing supplemental dietary antioxidants at it. We now know that the biology of reactive oxygen species is much more nuanced. For example, we know that our innate immune system requires reactive oxygen species in order to be able to defend off challenges. We also know that prolonged exposure to environmental stresses, and of course, aging itself can lead to cells and tissues that are chronically challenged with at least low levels of reactive oxygen species, and that this is different than we might see with reactive oxygen species challenges in more of a pulsatile fashion with physical activity, with exercise, which we know is a health span promoting intervention. So if you've followed the literature that's emerged over the last 15 years or so, there have been a number of groups really interested in understanding whether or not there's an interaction between supplemental dietary antioxidants and beneficial adaptations to exercise. And for the most part, it's been pretty evenly balanced between studies that have shown no unfavorable interactions that we can see or observe, with those that have said, yeah, it looks like dietary supplemental antioxidants may actually abrogate or squash some of the exercise-induced benefits. So this was the first study I'm going to share with you was a study that so many students got to participate in because it took a long time to do. And it was a study in which we took kind of a fundamentally different approach to asking this question about interactions between manipulating redox balance and exercise adaptations. And we focused on our favorite transcription factor, NRF2. This is a hugely oversimplified cartoon showing NRF2 bound to KEAP1, which essentially is a protein that helps to escort NRF2 to be degraded by the ubiquitin proteasome pathway in the absence of cell stress. That makes it so that it has a really short half-life, between 15 to 60 minutes, depending on the model organism. However, when there's cell stress present, what we know probably the most about are redox stresses. This allows NRF2 to escape its inhibition by KEAP1, enter the nucleus, and find the antioxidant response element that's present in more than 400 genes that I'll just collectively say are involved in adaptive homeostasis, meaning their job is to adapt to the stress and help the cells, tissues, and organisms reestablish homeostasis. This is a wonderful review paper if you're interested in this topic, where Dr. Davies and Pomato pretty exhaustively review the literature looking at a variety of stresses and across model organisms from cells through humans, showing that when we're young, or when your model organism is young, we have a very wide range of ability to adapt, a homeostatic range to adapt and reestablish homeostasis. But with age, this ability and the range that we have to adapt to stresses is much narrower. And since NRF2's job is very important in this adaptive homeostatic response, it may become most important with advancing age. In fact, it can be knocked out, and animals do quite well when they're young. So in our lab, we just kind of did this study, peeling animals off of other studies when they were perhaps ones that ran a little too well for the other studies that we were doing. So we used rats that ran many kilometers a night on their cage wheels. And our primary outcome was looking at this very well-known adaptation to exercise training, and that is muscle mitochondrial biogenesis. These two talented then trainees, now Dr. Bruns and Drs. Ehrlicher, took a lead on a lot of these projects, and they wanted to test the hypothesis that in these rats, treatment with vitamin C supplements would inhibit muscle mitochondrial adaptations to exercise. But if we treated them with NRF2 activators, we can talk more about that later if you're interested, but they're essentially synergistic phytochemicals that activate NRF2, that this would permit the exercise-induced mitochondrial adaptations to exercise. So we had 4 different groups. One was a group of rats running on their wheels on just a regular diet. One was supplemented with vitamin C beyond what they already biosynthesize, and mimicking those found in a number of those studies that I listed in those 2 columns on the previous slide. Or they were supplemented with a NRF2 activator. And then we had one group where we locked their wheels and made them sedentary. And our primary outcomes were focused on muscle mitochondria. And so we use a stable isotope of water to allow us to measure the rates of mitochondrial biogenesis over the course of the study, rather than relying just on markers of mitochondrial biogenesis. And so I'm just going to show you one piece of data from this study where we looked at, this is the plantaris muscle, which is activated well on running wheels. We measured other muscles as well. And the primary outcome here was looking at mitochondrial biogenesis. So looking at the fractional synthesis rate of new mitochondrial proteins. If we compare the sedentary rats to the control fed rats who were on their wheels, we see the anticipated increase in mitochondrial biogenesis. But if they were supplemented with vitamin C, this mitochondrial biogenic response to exercise was completely abrogated. However, if they were treated with the NRF2 activator supplements in their diet, this was permissive and allowed them to carry on with this mitochondrial adaptation to exercise. We fractionate our tissues, so we always get the other fractions as well. If you look at the cytosolic enriched fraction, the proteins in the cytosol followed the very same pattern to mitochondrial proteins. However, we thought it was really interesting and unanticipated that contractile proteins, so this is a fraction where it's mostly myofibrillar proteins, vitamin C did not interfere with this adaptation to exercise, suggesting that these are far less redox sensitive adaptations. So the take-home message from our study here would be that vitamin C supplements do seem to interfere with beneficial adaptations in the mitochondrial proteome, but that NRF2 activators are permissive for these beneficial adaptations. I don't have time to share with you some really fun data, so I wanted to just give a plug and ask you to watch for some new data that we have using NRF2 activators in a different model. This is a guinea pig model that develops very reproducible musculoskeletal decline in the first part of their life. And I'll give you a little spoiler here that treatment with the NRF2 activators indeed improved many aspects of musculoskeletal decline. We focused on cartilage, bone, muscle, and actually tendon too. And this is the talented team of people that will be publishing those studies. So indeed, if you look at NRF2 activators and exercise, both treatments protect against decreases in health span, and there's no evidence of unfavorable interactions. We're excited enough about these that we're currently doing a small clinical trial in humans with age-related mobility impairment. So now I'll move on to a published set of studies looking at interactions between exercise and metformin. And they kind of build on, long-standing knowledge from the diabetes prevention program that both metformin and, of course, exercise extend health span. This is well-known data where they looked at cumulative incidence of diabetes, comparing a group of people who were either treated with placebo, with metformin, or they were counseled to make some lifestyle changes, including increasing physical activity and dietary changes. And you can see that both metformin and lifestyle interventions decreased the cumulative incidence of type 2 diabetes compared to placebo. So with the many, many decades we have of experience with metformin, we know that it's a safe drug. It's a biguanide that's very frequently used for treating type 2 diabetes. So we know a lot about it. Therefore, we went straight to a human model to test this hypothesis. This was Dr. Adam Konopka's work when he was a postdoc in my lab. And his hypothesis was that metformin would accentuate the positive adaptations to exercise, as observed by greater improvements in skeletal muscle mitochondrial respiration, insulin sensitivity, and cardiorespiratory fitness. So this was a pretty straightforward study where we did pre-treatment testing and post-treatment testing that included OGTTs to look at glucose tolerance, VO2 max testing for cardiorespiratory fitness, and then we took muscle biopsies to enable us to look at muscle mitochondrial function. And our older adults who were sedentary and had at least one risk for type 2 diabetes, it's actually pretty hard to recruit people with type 2 diabetes in northern Colorado. They came in for a supervised aerobic exercise training program in which they either received placebo or what we would consider to be a clinical dose of metformin when used for people with type 2 diabetes. And then we did all the same testing over in the end. I'm going to show you 3 pieces of data from this study. And I'm going to show them all to you as a delta, the change from pre to post exercise training. The first one being results from the oral glucose tolerance test, looking at whole body insulin sensitivity. And what you see in the placebo group here is that pretty much all of the participants, they either stayed the same or they improved their glucose tolerance. If you look at this, the first thing that should jump off the screen that's different about the metformin group is how variable this group is. This was crossing my mind during our first talk of the day, focusing on variability in the exercise response. So about 50% of the people taking metformin actually had a decrease in their glucose tolerance or their insulin sensitivity, while the other half either stayed the same or improved. If we look at cardiorespiratory fitness, our findings here are not significantly different, but I think they're important because we know that VO2 max is one of the best predictors of all-cause mortality in people. So if we compare the metformin treated group, they had approximately half the improvement in cardiorespiratory fitness compared to placebo with exercise training. Last piece of data from the study. This is an ADP titration protocol that we did in permeabilized skeletal muscle fibers. And again, I'm showing you these as a delta, a change from pre to post training. So if we look across all of these ADP concentrations where we were trying to hit on some submaximal all the way through maximal concentrations, you can see in the black bars, the placebo group had increases in oxygen consumption with exercise training across all of the ADP concentrations that were completely absent with the metformin treated group. So contrary to our hypothesis, metformin did not accentuate the effects of exercise training on insulin sensitivity, cardiorespiratory fitness, or muscle mitochondrial function. Dr. Barry Braun, our department head, led a really nice series of studies when he was at UMass, showing that metabolic syndrome outcomes and cardiovascular disease risk were also not improved with concurrent treatment with metformin. In fact, our collective data show that metformin may blunt some of these beneficial adaptations to exercise training. So while metformin is an excellent drug for type 2 diabetes, we think that there's still quite a bit of work to do to understand this cost-benefit ratio, particularly in people who are very likely to adhere to an increase in physical activity. Last group of data I'm going to show you is a recent study published by a postdoc in my lab, looking at SGLT2 inhibitors in exercise. So SGLT2 inhibitors are another class of compounds used to treat type 2 diabetes. Stands for sodium glucose cotransporter 2, which, if you're not familiar with these transporters, they sit in the filtration apparatus of the kidneys. So our kidneys filter in an adult about 180 liters of plasma per day, which equates to about 160 to 180 grams of glucose. And these transporters are responsible for reabsorbing about 90% of that glucose back into circulation. So it should make sense that if we inhibit those with drugs, that we're going to spill more glucose into the urine. And that's the mechanism of action for these drugs that are pretty good at treating type 2 diabetes. So Melissa Linden, when she was a postdoc in my lab, wanted to address the hypothesis that when SGLT2 treatment, and we used one of the three drugs approved by the FDA in the States, canagliflozin, when it's combined with exercise, there will be a further improvement in glycemic control compared to exercise or drug treatment alone. And importantly, she thought that SGLT2 inhibitor treatment would not attenuate the exercise adaptations like we saw with metformin. So we went back to the rat model for this, where we either had rats running or sedentary. This is a single piece of data shown in two ways. I'm only going to show 3 pieces of data from this study. So with a glucose tolerance test, we measured the glucose area under the curve. This was a rat model, as I mentioned, and to induce type 2 diabetes, we fed them a high-fat diet. So that's what's happening with all of these groups. This is the only one not fed a high-fat diet. And then we treated them with a very low dose of streptozotocin. So they didn't have complete beta cell failure. They continued to make insulin. And you can see the model was successful in the vehicle-treated sedentary animals. We induced glucose intolerance, as you can see, with an increased glucose area under the curve that was improved with exercise training, also improved with canagliflozin treatment. And then the greatest improvement was when we combined those two, SGLT2 inhibitor treatment and exercise. So the take-home message from this is that canagliflozin plus exercise further improved glucose tolerance compared to either mono treatment. Here are the last data, and then I'm done. So we had relatively few runners by the end of the study. I'm only showing you animals that completed the exercise training and were either treated with the vehicle or the SGLT2 inhibitor. You can see the sample size is small. We've repeated this since that time, so we know that it's a consistent finding. So what you're looking at here is, well, we were running the rats at a pretty low intensity, VO2 max, or 50% of their estimated Vmax. If we measured their respiratory exchange ratio, what we saw was that there seems to be an increased reliance on fat for energy with SGLT2 inhibitor treatment. If we popped up their intensity to about 75% and ran them till they were exhausted and looked at the distance traveled, first you'll see none of them traveled very far, but the ones treated with SGLT2 inhibitors ran about twice as far before they were exhausted. So the take-home message here is that while metformin interferes with adaptations to exercise in our human model, in this rat model, the SGLT2 inhibitor canagliflozin had a permissive or even additive interaction with exercise. So obviously this needs to be investigated further in different populations of people, as well as with other SGLT2 inhibitors, but it seems like it's worth pursuing SGLT2 inhibitors as an alternative to metformin, particularly in people who are very likely to adhere to an exercise training program. So I hope that you'll agree with me that it's worth putting some effort into looking at interactions between health span extending interventions, particularly with exercise training. And in light of some of the things that we've been talking about in this panel, that everybody responds to exercise a little bit differently. This is my beautiful group of people. We have not been in the same room together in a very long time, and now I've lost a bunch of them to graduation, so I'm starting over. I've named most of the people who were involved in collecting these data. Big thanks to the panel organizers and to people who funded this work.
Okay, thanks so much. Great talk. We have a few questions popping up here. The first one from Michael Bohamdan. Have you ever checked A1C level?
I have not in the animal study. Yes, I have actually. In the canagliflozin study that isn't published yet, we measured A1C. Hard pressed to remember if we had any treatment differences. We know that we increased A1C with the STZ and high-fat diet in those animals. I'm hard-pressed to remember if we improved it over the relatively short course of the exercise and cana treatment. As we know, A1C is, in humans anyway, reflective of about 120 days of glucose control. In our human study with metformin, exercise training improved A1C in almost all of the participants, and that was not inhibited by metformin.
Okay, here's one from Nicole Riddle. Would you mind commenting on the vitamin C supplementation study and the impact you might be having on DNA methylation, since vitamin C can induce TET function?
My comment would be, let's talk about that. Yeah, it's a possibility. So I think there are so many different ways that vitamin C could interfere, particularly in the doses that are used both in those human trials and animal trials where we're interfering with exercise adaptations. Also, remember, in a rat, it's not a great model, which is why we've kind of shifted towards guinea pigs for some of this work. Guinea pigs don't biosynthesize vitamin C, and so we're much more in control of the amount of vitamin C that their DNA is exposed to. So that's about the biggest comment that I can have, but I'd love to continue that conversation.
Okay, and we have one more question. Have you tried NRF2 activators without exercise, and do they act as exercise mimetics?
That's a good question. So we are trying NRF2 activators, and by we, I mean that one of them has has made it through the NIA interventions testing program, where they look at lifespan extension in a heterogeneous mouse model. And it increased median lifespan in the male mice, but not the females. We have a kind of second generation of NRF2 activator in a current cohort of the ITP. So preliminary studies there aren't looking as promising as the first NRF2 activator. What we haven't seen, though, haven't really tested in a well-powered way, is that treating with a NRF2 activator all by itself can stimulate mitochondrial biogenesis or increase cardiorespiratory fitness or exercise capacity. So I don't think it's the magic pill to get all the beneficial adaptations to exercise. But there is some known crosstalk of NRF2 with mitochondrial biogenic pathways. We had actually, in that rat study, hypothesized we might see an additive effect with exercise and NRF2 activation in terms of mitochondrial biogenesis, and we didn't see that.
Okay, we're gonna move on to the final talk of this session. So Laura, if you wanna go ahead and start putting up your slides while I introduce you. Laura Reed is coming to us from the University of Alabama in Tuscaloosa, and she has been studying population genetics and how genetic and environmental factors interact to shape metabolism. And she has also been interested recently in exercise in the fruit fly model. And so we're excited to hear about what she's been doing in that area. So go take it away, Laura.
Thank you so much. It was really exciting to be a part of this. And I want to work on expanding some of what Dr. Riddle mentioned at the beginning of this talk, and talk a little bit more about how we might be looking at variation among individuals or among genotypes and how that can be impacting the response to interventions like exercise. So this is sort of a summary of my worldview of biology. So when we're thinking about the phenotype of an organism, such as something like its response to exercise or body weight, obesity, there are many levels that contribute to that ultimate phenotype. We have our genetic component from the genome. That genome gets modified by epigenomic factors. And then there's the transcription process, the proteomic or proteome space where the proteins function, which then in part act on the metabolome, producing the final phenotype. And then there's, of course, feedback mechanisms among all of these internal, or what I call endophenotype levels, to produce the final phenotype. But what often happens is we sort of forget about the factors that influence how these levels interact with each other, and thus might ultimately contribute to phenotypic variation. So part of what we're definitely talking about here is how things like exercise influence ultimate phenotypic variation in terms of outcomes in health span, for example. And we need to think these environmental factors interact with many different potential levels within this endophenotype spectrum. And the regulatory feedback mechanisms are part of what help maintain the homeostasis to produce the final observed phenotype. The other thing we tend to forget to pay attention to is the natural genetic variation. So the way that natural genetic variation is funneled through these different endophenotype steps to produce final phenotypic variation is an area of fascinating interest, but challenging to study, because controlling both genetic variation factors and environmental variation factors simultaneously, especially in an organism like humans, is really challenging. So as we are all quite aware, we do have a concerning issue with obesity in the US and honestly worldwide, with increasing rates of obesity throughout the US. Karyn, that you're mentioning that it's hard to find diabetic recruits in northern Colorado. It's pretty easy to find them around here in Alabama. We're in the southeast area where we have exceptionally high rates of obesity and type 2 diabetes. So what we know about these traits from the genetic perspective in humans is that there's nearly 3,000 genetic associations for obesity in human populations. So that means we know that there are at least 3,000 different positions in the genome that can vary and play a role in shaping obesity, the obese phenotype in a human. But what that also means is that despite there being a significant or substantial genetic contribution to the obesity phenotype, only a tiny portion of the genetic variation can be explained by any given variant. So it's hard then to say which individuals are going to be responding in which ways to potential interventions, in part because there's such a wide range of possible ways we could be shaping their phenotype from the genetic perspective. So I endeavor to try to wrap my head around how these factors actually interact with each other. So in this example, we have a wild population. So this could be a wild population of humans, or in my case, fruit flies. And here the phenotypes for the individuals in that population are represented by different colors. So one thing we can do in a research lab with an organism like fruit flies is we can establish these inbred lines that Dr. Riddle mentioned and robustly estimate the genetic variation among those lines for a given phenotype. So that leads to a fairly strong statistical estimate of the genetic component of variation that we might observe in the wild. But remember, also in the wild, we have that environmental heterogeneity influencing these organisms. Diet, exercise, toxins, whatever might be in their environment. And that's where it gets really sticky to try to analyze this in something like humans. But in flies, we can systematically test those different genotypes across all the different environments we might be interested in, and then robustly estimate how the environmental variation and the genetic variation interact with one another. So I just want to mention briefly about the life cycle of the fruit fly. This is relevant because it fits into the experimental design I'm about to tell you about. Most of the studies done in my lab focus on these larval stages. So the flies spend about 4 days developing through from first instar to third instar and then pupate. So most of what I'm doing in the lab is actually manipulating the environment of these larvae, primarily by diet, and then I'm looking at some other factors too. But then the exercise component of what we do is done on the adults. So we can manipulate the diet stage and then look at the diet effect on adult exercise phenotypes. So the basic design of any one of my studies basically starts out with we have multiple genetic lines. We raise those in multiple environments. So in the examples I'm going to show you today, those environments, dietary environments, are a normal diet, so what we would consider sort of a healthy lab diet for a fruit fly, and a high-fat diet. And then we measure the phenotypes that they produce. There are many possible ways the flies could react to that experience. So these are just a few hypothetical examples. So one possibility is that every genotype responds or doesn't actually respond to this intervention at all. And so we see no variation in either genetic or environmental responses in these two genotypes where the lines are horizontal and parallel. But if there's genetic variation, we'd see a difference between the two lines, but that difference would not interact with anything about the environments. A purely environmental response would show a change with environment, but no difference between the genetic lines. And then finally, there's a possibility of an interaction between genetic and environmental factors, where hypothetically, in this case, the red genetic line has a decrease in the measured phenotype in environment 2 relative to environment 1, while the blue genetic line has an increase. So it turns out, perhaps not surprisingly given my title, that those kinds of interactions are a major factor that's going on in these wild populations, or the populations of flies. So this is looking at just one phenotype we consistently measure, with that's total triglyceride content in the body of the fly. And if we compare the triglyceride content in the fly that's been raised on a normal diet, so these are larvae that have been raised on a normal diet versus a high-fat diet, but of the same genotype, there are some genotypes, like those that are coded in red here, that show an increase in triglyceride storage when they're eating a high-fat diet, But there are also others that are the blue here that show a decrease in triglyceride storage when they're eating a high-fat diet, and a few that don't respond one way or the other. And so we're seeing both genetic and environmental factors interacting in producing the phenotypes we observe. On the right side, what we're looking at is how that variation is partitioned across these different factors that I'm able to quantify. So the gray bars here are indicating the proportion of the total variation that's directly attributed to only genetic factors. The white bars here are right portion is the proportion that's only environmental, only due to diet. And then the black portion is the portion that's actually this interaction effect. So for triglycerides, blood sugar, weight, other phenotypes that we measure, we often, almost always see that the dietary component is much smaller than either the main genetic effect or the interaction effect, which to me, when I first was analyzing this data, was a big surprise. I was expecting there to be a much larger proportion due to diet alone. So that led us to really focus on what's going on in that interaction portion. So, so this is a setup to tell you sort of like the basic design of many studies that are done in my lab. And then I want to introduce our contraption. So Dr. Riddle mentioned this briefly. So this is the tread wheel. So what it does is it slowly rotates and the flies within the tread wheel experience a constantly changing direction for what is up. So they use that negative geotaxis characteristic where they like to be at the top of things. So when we slowly rotate the vial, the flies basically just keep climbing and we're able to induce a significant amount of exercise through that. So the basic design we use is the vials that the flies are contained in are either our exercise vials where there's 6 centimeters of movement space for the flies, or our controls where there's only 1 centimeter of movement space so that they are essentially forced to be sedentary while on the inducing experience of the tread wheel. We start out with 3 to 5 day old adults and exercise many genetic lines. And the basic design we use is an alternating exercise rest sequence for up to 80 minutes in a day. So on day 1, they get 15 minutes of exercise and 5 minutes of rest, alternating for a total of 60 minutes of exercise. And then by day 5, they're up to 20 minutes of exercise with 5 minutes rest and a total of 80 minutes of exercise. So it turns out that exercise does work in flies, as we've seen from others as well. So this is looking at across multiple genetic lines. Overall, we see that there's can be a substantial decrease in stored triglycerides in adult flies who have experienced exercise relative to controls, a decrease in body weight as well relative to controls, and an increase in protein. We also have measured things like glycogen stores. Those also overall tend to decrease in the context of exercise. And we see an improvement in climbing performance. So the flies are better at climbing rapidly following this exercise treatment than if they had the sedentary treatment. So that's when we look at these genotypes overall, sort of averaging across all the genotypes. But when we actually dig in a little bit, we see that the story is more complex, as Dr. Riddle implied. So this is looking across the 4 particular genotypes this particular study was tested against. So these are 4 different wild type lines and actually ones that are commonly used in fruit fly research labs. And what's being graphed on the y-axis here is the change in the phenotype in response to the exercise treatment. So if it's negative, it was a decrease in phenotype. If it's positive, it's an increase in phenotype. So we find that, for example, here with the triglyceride phenotype, that we see a fair amount of decrease, but only significant decrease in 2 of the genetic lines and only in the females. So the overall, what we're observing as an overall decrease in triglyceride storage is really being driven by the 2 genetic lines and the females of those 2 genetic lines. Similarly with climbing, where we see that there are many examples of where there's improved performance, increased climbing performance, but that's not uniform. That's not all genotypes. So the Oregon R genotype, which did have a substantial decrease in triglyceride storage, did not change in performance in terms of its climbing ability. And then for like this genotype on the far right here, the females improved in climbing mobility substantially following exercise, whereas the males did not show a significant increase. So we see a similar kind of interaction effect involving genotype and sex for the other phenotype to measure weight, proteins, total body protein, glucose, and glycogen stores. Okay, so we've established that there are complex interactions among the genotypes and with how they respond to exercise. And earlier I showed you there's complex interactions between genotype and diet. And so the next step we wanted to take is what happens when we combine those two. So in this study, we're taking those multiple genetic lines and then raising them either on a normal laboratory diet or a high-fat diet as larvae. So in those first 4 days of growth and development. And then we partition them out by sex and do the control and exercise treatment for each of those sex and dietary treatments in the adults. All the adults are eating a normal healthy lab diet as adults. So we've only exposed them to the dietary intervention as larvae, and they only get healthy food, well, what we hope is healthy food for the flies as adults. So in this, we are really trying to isolate what factors or to what extent can adult exercise mitigate any impacts of a larval diet. Perhaps having some relevance to what we observe with pediatric obesity, for example. We see elevating rates of pediatric obesity. Is it possible that individuals that were obese as children have the potential to recover some of their health with a dietary intervention or, an exercise intervention as adults? So basically, the answer is exercise works better for some than others. So the adult exercise impact interacts with both the larval diet effect and the genotype effect. So this starts looking a little complicated, so I want to just walk you through it so you can understand what's going on. So on the left side here are the flies that did not receive exercise. They were the control, no exercise as adults, no induced exercise as adults, and whether they had a normal diet or a high-fat diet as larvae. And so, for example, this red genotype here had a substantial decrease in its climbing performance if it was raised on a normal diet relative to a high-fat diet. So the high-fat diet as a larva decreased its performance in the absence of exercise. But when that same genetic line was allowed to exercise after having a high-fat diet, it actually saw an improvement relative to a normal larval diet in the presence of exercise. So its climbing increased. So there's some genotypes where that exercise intervention looks like it could be really beneficial, but for others, not necessarily. So there's some here where the exercise intervention for this particular line, the yellowish color, actually showed a decreased performance relative to a normal diet with the exercise treatment, and really no difference relative to control. So that's the line there. Its phenotype, its climbing performance is middle of the road on the control treatment and on the exercise treatment when it's eating a high-fat diet. So basically, it gets complex. So what I wanted then to bring this back around to is how do we then wrap our heads around this complexity? If there's so many factors that contribute in a nonlinear fashion, it quickly gets overwhelming to think about how do we make general recommendations that might be helpful. And so one of the other aspects of what my lab does a lot of is metabolomics. So we measure the complete metabolome, that is, or as much as is experimentally possible, of the small metabolites in an organism. And this particular data set is measured on larvae, and larvae that were fed different, in this case, 4 different diets, including a normal diet and a high-fat diet. And what we found is that if we looked at the metabolomic profile as a whole and then performed principal components analysis on it, we found I don't know why it was the second, but the second principal component was very good at predicting a number of the phenotypes in the larvae. And so that is when other factors like gene expression level and other things like that were not particularly helpful. They did not do a particularly clear job of predicting across dietary interventions and across genotypes. So it turns out the second principal component of the metabolome was quite valuable. And the metabolites I have listed there are the ones that had the heaviest loadings on that principal component. So L-DOPA and arachidonylyl dopamine being neurotransmitters or neurotransmitter precursors. And then we also have things like the branched chain branched chain amino acids that have shown up as good biomarkers for other aspects of obesity and diabetes in humans. So there's a possibility that the metabolome might be a good indicator that can be more generalized than perhaps genetic factors or gene expression factors for looking across genotypes and across environments. On the right side, we collaborated with Rolf Bodmer's group to look at heart function in these flies. And in this case, these flies, the particular flies that were measured, did not receive a dietary intervention. They were just doing their normal diet conditions. And then we asked whether what their phenotype response was to dietary factors, whether that correlated with any aspects of their if their heart performance as adults correlated with any aspects of their larval or in this case pupil phenotypes. And basically what we found is that the arrhythmia index, so the development of arrhythmias in adult flies, did correlate with both phenotypic factors and also correlated with this first or second principal component of the metabolome. So we're actually able to use the larval metabolome to predict adult onset heart arrhythmias. So that's an exciting potential direction that we are going to be exploring further. So basically the take home is that metabolic phenotypes are shaped by complex interactions of genetic and environmental factors. So here I've shown you exercise, diet, sex, and genetic variation. Our lab is also looking at factors like sleep and the gut microbiome. And metabolomics can be helpful in potentially identifying biomarkers that could be more generalizable across environments when other endophenotypes perhaps are not as reliable. So I just want to highlight that most of the exercise work you saw presented here was started by Sean Mendez and then continued by Kelsey Lohman and Roland Wyatt here in my lab. And then many additional undergraduates contributed a lot to it, and my collaborators and funding sources. So thank you very much.
Okay, thanks, Laura. We have a couple of questions up here. One from Pat Miller. How does your design account for time versus age effects? In other words, the different environments occur over time, but may depend on at what age they start and finish, particularly in females. So for the dietary treatment specifically in that window as larvae, which is just that four day or so window. For the adults, the exercise interventions we've done thus far focus just in those starting with 3 to 5 day old adults and only going for 5 days following that. So we haven't really looked into the longer term impacts of these exercise or well, to some extent we've done diet, but not as so extensively in terms of the longer term impacts. So basically we're concentrating on that sort of narrow time window for the interventions. And here's another one from Girish Haranath. Did you look at the influence of exercise on ketone metabolism and if this metabolic shift may be playing a role in physiological benefits, for instance, through metabolomics or gene expression of key enzymes BHB?
So we have not, not directly or not specifically, but ketones not pop out as well correlated with much of anything in terms of our metabolomic analysis. So they didn't raise any flags as being interesting in that dimension.
I have one more question from myself. With regard to the male versus female differences, have you ever put any of females that have a reproductive mutation on the tread wheel to see if you subtract the reproductive element, if the difference changes or goes away?
We haven't tried that, but I think that would be really interesting. We are careful to make sure that the females are mated So everybody's had the opportunity to mate, because we also know, of course, that there's significant metabolic impacts of just manufacturing eggs. So we make sure that everybody has had the same experience, but no, we have not tried taking reproduction out of the equation yet.
Okay, cool. So we're actually, we've run behind schedule. So we were normally going to have another general discussion section that was going to end at the bottom of the hour, but we're already past the bottom of the hour. So that's it formally. Well, I'll stick around for a few minutes. If Elena can stick around for a few minutes, just to make sure there are no more questions or discussions. And then that'll be it until tomorrow. And I will really look forward to seeing all of you back. And tell your friends, if you have other friends you think would be interested in tomorrow, I'd be happy to broaden it out with more participants tomorrow. That would be fantastic. Thanks, everybody.
Yes, before we go, I would like to thank you, Robert, for chairing this session and all the speakers.
Right, wonderful. Thank you all so much.
Okay, bye-bye.